everyone, welcome back once again in your biology class. This is Nowsin and you are watching Educational Support. If you like this video, then share it with your friends, and if you want more videos, then subscribe this channel. Do you have some minutes? Today I gonna teach something to you, that is digestion and absorption of carbohydrates. This is the second half of this video. The previous one was focused on the digestion of carbohydrates, but this one will be talking mostly about on the absorption of carbohydrates. So let us learn more in detail about absorption of carbohydrates. In this video, we are going to focus on this topic. So let's get begin. Do you know what is absorption? Absorption is the process of the absorbing or assimilating substances into the cells or across the tissues and organs through the process of diffusion or osmosis. The principal monosaccharides produced by the digestion of carbohydrates are glucose, fructose, and galactose. Glucose accounts for the 80% of the total monosaccharides, and only monosaccharides are absorbed by the intestine. The absorption occurs mostly in the duodenum and upper jejunum of the small intestine. So absorption rate is maximum for galactose, moderate for glucose, and minimum for fructose. Now we are going to discuss on absorption rate. Do you know what is Cori study? Yes, Cori studies the rate of absorption of different sugars from small intestine in rat. Glucose absorption as 100, comparative absorption of other sugars as galactose 110, glucose 100, fructose 43, mannose 19, xylose 15, and arabinose 9. So galactose is absorbed more rapidly than glucose, and absorption rate is maximum for galactose, moderate for glucose, and minimum for fructose. So different sugars passes different mechanisms for their absorption. Monosaccharides are the end products of carbohydrate digestion, enter the capillaries of the intestinal villi. Glucose, fructose, and galactose are absorbed across the membrane of the small intestine and transported to the liver, where they are either used by the liver or further distributed to the rest of the body. Next, we are going to focus on mechanism. Carbohydrates are absorbed as monosaccharides and absorbed by three basic mechanisms, namely passive diffusion, facilitated diffusion, and active transport. Do you know what are the characters of passive diffusion? The characters of passive diffusion are high to low concentration, no energy is required, no carrier protein is required, and the last one is it is the slowest process. This is the facilitated diffusion. So the characters of facilitated diffusions are high to low concentration, no energy is required, but carrier protein is required, and the last one is it is the fast process. Next is active transport. So the characters of active transport are low to high concentration, energy required from ATP, carrier protein is also required, and the last one is it is the fastest process. Now we are going to focus on glucose transporter. There are two types of glucose transporters for transport of glucose. Number one is sodium dependent glucose transporter, also called SGLT, and number two is sodium independent glucose transporter, also called GLUT. So there is an important note that glucose is a polar molecule, so it cannot pass through lipid bilayer. So first is sodium dependent glucose transporter. Do you know what is sodium dependent glucose transporter? It is a form of co-transport. Two binding sites on the transporter, one for sodium and other for glucose. Sodium binding is important. Do you know why sodium binding is important? Sodium binding is important because it binds with sodium firstly, which causes the structural changes in the transporter which allows glucose to bind. Sodium is transported across the cell membrane from higher to lower concentration, and glucose is transported against concentration to the proximal tubule epithelial cells. From ATP is used to pump sodium out via the sodium-potassium ATPase pump, and glucose diffuses down its concentration gradient into the interstitium. Both glucose and galactose are absorbed by sodium-dependent process. Do you know where SGLT1 and SGLT2 are present? This picture showing that SGLT1 is present on the intestinal cells, where SGLT2 are present on the proximal renal tubular cells. Do you know what are the reasons for expulsion of sodium? Yes, there are three reasons for expulsion of sodium. Number one is sodium is osmotically active, which causes osmotic flow to cells and leading to osmolysis. Number two is sodium is inhibitory to many enzyme actions. And number three is sodium concentration has to be kept minimal to maintain the downward gradient. Next, we are going to focus on sodium independent glucose transporter. Do you know what is sodium independent glucose transporter? This sodium independent glucose transporters are used for facilitated diffusion. They are numbered from GLUT1 to GLUT14. For example, GLUT1, GLUT2, GLUT4 and GLUT5. So, GLUT2 and GLUT4 are very important. Now, we are going to discuss on GLUT1. It is the present in RBCs and brain. It is also present in retina, colon and placenta. It helps in glucose uptake in the most of these tissues, which is independent of insulin. Next is GLUT2. GLUT2 operates in intestinal epithelial cells, and it is a uniport facilitated diffusion system and not dependent on sodium ion. Glucose is held GLUT by weak hydrogen bonds, and after fixing glucose changes configuration and opens inner side releasing glucose. Do you have any idea what is GLUT4? GLUT4 operates in the muscles and adipose tissues. So it is under control of insulin. And insulin that induces the intracellular ZLUT4 molecules to move to the cell membrane and increases the glucose uptake. Next, we are going to focus on absorption of fructose. Fructose absorption is simple and does not require energy and sodium ions, transported by facilitated diffusion mediated by a carrier. Inside the epithelial cell, most of these are converted to glucose and the latter then enters into the circulation. So next is factors that affect absorption of monosaccharides are mucous membrane is not healthy, absorption will decrease and the absorption is faster through intact mucosa and the absorption is decreased if there is some inflammation or injury to the mucosa. Next is thyroid hormone. Thyroid hormone increases the rate of absorption of glucose. It increases absorption of hexose and acts on intestinal mucosa. Mineralocorticoids that affect absorption of monosaccharides and that is aldosterone. Aldosterone increases the rate of absorption. Number 4 is vitamins. Vitamin B6 and B12, pantothenic acid and folic acid are required for absorption of glucose. Absorption is decreased in deficiency of vitamin B complex. That's all of my presentation. I hope you enjoyed this lecture as much as I enjoyed putting it together for you. If any inquiry, then put down a comment under this video and also press the subscribe button and bell icon right next to it and give us a thumbs up and happy watching. Thank you.